Nucleic acids, AQA biology. Key terms. So you have less than the other videos actually for nucleic acids. And the main key terms are, what is a nucleic acid? Well, it's a polymer made up of many nucleotides. 5 prime, which is the top of the molecule, so the DNA strand, and then 3 prime, which is the bottom of the molecule. You have mRNA, which is single strand RNA polynucleotide chain. tRNA, which is a single strand RNA chain folded into the shape of a clover, and it has an anticodon at the top, at the bottom, and an open end at the top, which combines to amino acids. However, that will be more into chapter 8 of protein synthesis. And RNA, which has 2 subunits, a single strand of RNA variously folded and twisted, forming a tertiary structure. That's more, we'll go on to that in chapter 3 about organism, organelle shapes and purposes and such. And then semi-conservative replication, which is the way in which DNA is replicated and multiplied in a reaction. Structure of RNA and DNA. Now, the basic bonding in an RNA or a DNA molecule is that you have a base bonded to a sugar, which is formed by a glycosidic bond, and a phosphate molecule binding to the sugar as well, formed by an ester bond. Now, this is done in a condensation reaction. Now, the 4 main bases in DNA, or the only bases, is adenine, guanine, cytosine, and thymine. You have different length nucleotide chains, and if you watch my previous videos, then you know about the abbreviations in front and what they mean. So you have mononucleotides, dinucleotides, polynucleotides, and so on. Structure of a nucleotide in the helix. Well, DNA, as it's a double-stranded molecule, it runs anti-parallel. So basically, you'll have the original strand and the replicated strand, but they run in opposite directions. And as they run in opposite directions, it means that the bonds that form between the bases are all complementary to each other. For instance, adenine will bind to thymine, guanine to cytosine. And the bonding relationships are purines, which is adenine and guanine, to cytosine and thymine, which are pyrimidines. Sugar and deoxyribose sugars. Oh god, that's wrong. Sugar's in DNA, and the sugar that's in DNA is deoxyribose sugar. Sorry about that. Now, the main function of nucleotides is the formation of DNA and RNA. Now, how is DNA designed for its function? Well, it's thermally stable, and what this means is that it can withstand high temperatures without deforming and denaturing. It has a rigid backbone structure of a pentose and a phosphate molecule, which are held together strongly by phosphodiester bonds. All bases are complementary to the other in the other strand. So cytosine will only bind to thymine, cytosine will only bind to guanine, and adenine will only bind to bond to thymine, sorry. Jeez, that's a hard word to say. All bases are held in place by hydrogen bonds and disulfuric bonds, and bases are easily split by enzymes to allow replication. Structure of RNA molecule. An RNA molecule is formed using a ribose sugar, a phosphate, and RNA bases. Now, RNA bases would be the same as DNA, so adenine, guanine, cytosine, but instead of thymine, it's uracil instead. And it's a single nucleotide chain, and the arrangement is important to its function, which we'll get into in protein synthesis. And the different types of RNA are mRNA, tRNA, and rRNA. DNA replication. So these are the main steps for DNA replication. So your DNA helicase unwinds and unzips the DNA, breaking the hydrogen bonds, forming two separate strands. Nucleotides attach to the complementary bases on the original strand. DNA polymerase joins the DNA nucleotides together, and then winding enzyme winds a new strand, forming double helicase, which will be the double helix. And this is identical to the original molecule. Now, there are three main ways that DNA can be replicated, but you only really need to know one for your exam. And the one that you need to know is semi-conservative replication. And this is how it occurs. So you have generally the example that's going to be given is about nitrogen 15 and nitrogen 14. So they'll be like, oh, nitrogen 14 or 15 is growing in a culture of nitrogen 14 or 15. Then they'll say that they're going to pour one strand, let's say nitrogen 15, into a culture of nitrogen 14. And basically you would obtain two bands. As nitrogen 14 and nitrogen 15 are different densities due to its mass number, nitrogen 15 is heavier, so it'll be at the bottom, and nitrogen 14 is lighter, so it'll be at the top. Now, in the beginning, you'll probably have a 50-50 split of nitrogen 15 and nitrogen 14. However, as it's being replicated, as it's growing in a culture of nitrogen 14, there'll be less and less nitrogen 15. So you have 50-50, then nitrogen 15 will go to 25 to 75 for the nitrogen 14. Then the next time it might be 12.5 and 85.5 and so on. Now, thank you for listening, and the next video will be completing the nucleic acids topic. So it'll be about ATP, water, and inorganic ions. Thank you for listening.